This is part 3 of lecture 1 on the cardiovascular system. So if you haven't watched the previous 2 parts, I'd go back and watch those first. This will make a lot more sense. This video is going to focus on the microvasculature. So when we're talking about the microvasculature, we're going to focus really on the capillaries, perhaps some mention of the other components of the microvasculature, and I will also then discuss the lymphatic system and how it interacts with the cardiovascular system. Now, capillaries are small. In fact, capillaries are really tiny. They're only about 4 to 6 microns in diameter, so barely big enough to allow a red blood vessel, a red blood cell or an erythrocyte to flow through. There's an example of one there. They're normally quite short as well, less than 1 millimeter. Now, they compose, if you remember, as a single endothelial cell layer, and they are the major site of gas and solute diffusion to and from tissues. And this can occur by transcellular exchange, where things pass directly across and gas is just diffused directly across the smooth muscles, across the endothelial cells, but also facilitated diffusion across the endothelial cell layer as well. We also get paracellular exchange, that's between the cells, and I'll mention some of these in detail over the next few slides, and also as part of vesicular transport, where vesicles might be transported across the endothelium. So here's just a couple of pictures to highlight blood, and again this is in the capillary network across a small section of muscle. And if we dive into that in a little bit more detail, we can see here the capillary wall, single layer of endothelial cells. You can barely see it. These are the muscle tissue underneath, and it's just large enough to pass through an erythrocyte. And we can also see a white blood cell there as well. So we've got to think about what capillaries do in the solute exchange and how they link to the rest of the cardiovascular system. Well, it is the terminal artery, so you get the arterioles, and it's the terminal arterioles which determine the number of perfused capillary beds. So this is the capillary bed here. This is the terminal arteriole feeding into that capillary bed. Now, in some blood vessels there are, or in some organs there are capillary sphincters which can shut off the blood supply to certain branches of the capillary network. It doesn't happen in all organs or tissues, and not all have these what we call meta arteriole, which basically vascular shunts. But it's often the way that we see it portrayed in the textbook, so it's worth knowing that this does happen as well. But normally it's the arteriole and the venule at the other side. The blood flows through the capillary bed, and it's just a large surface area, cross-hatched in nature, and that gives you a large surface area for gas and solute exchange. And then the blood is returned to the venules and then returned to the venous system. So by far the most abundant type of capillary are what we call the continuous capillaries. And they're just shown here because their appearance looks continuous, and between the cells there's very little gaps. Now, most of the diffusion that occurs is of gases and small lipophilic molecules. So gases and small lipophilic molecules, small fatty molecules, can just diffuse immediately across here and into the surrounding tissues, and they can diffuse for up to 300 microns. Now, there's not so much solute exchange in continuous capillaries. There is some, and it can occur via intracellular clefts or junctions, very small gaps in between between the cells, and they would form along these small gaps. So you can see there's not very much room for solute transfer, but some does occur. We also have caveoli. So caveoli, I'm just going to highlight here. And if we look at this in the progression, the caveoli will then form vesicles which can transport across and take molecules from one side of the endothelial cell to the other. So that's another way that we can get vesicle mediated transport. Not a great source of solute exchange, but certainly how proteins and larger molecules cross. And we also get facilitated diffusion. And I think Dr. Tamagnini has talked to you about facilitated diffusion, and that's where certain things such as sugar molecules are transported across the endothelial cell layer using special transporter proteins that normally use the ionic gradients as a power source, or the action of ATP as a power source for that facilitated diffusion. So as I say, this is the most common form. In some tissues, particularly those around the brain, there are a lot of tight junctions. Now, if you go back to my lectures on cells in the environment, I mentioned tight junctions in that lecture as well. These are very tight junctions, and you can see, unlike the cleft, which is over here, in this little cartoon, you'll see that it's actually pinched tight and closed, and that means really limits solute exchange and even the exchange of water between these tight junctions. So you can really prevent water flow in tissues. The next type of capillary are the fenestrated capillaries, and these are much more permeable to water and salts, and that's because they have a number of what we call fenestra. So fenestra are small windows, if you like. It's Latin for window, fenestra. Also, any Italian speakers out there will understand the word fenestra, meaning window as well. And in these fenestrated capillaries, we get small water-soluble molecules can also penetrate. So fenestrae are generally about 50 microns in 50 nanometers in diameter, and they can be closed by the fenestral diaphragm. So there is some control over solubility or movement of water, and that is done by the fenestral diaphragm. Now, I will point out, because most capillaries are not fenestrated capillaries and are continuous capillaries, by far and away the largest control of solute transport in the entire body is controlled by tight junctions, not by the fenestra. Because the fenestra found in fenestrated capillaries are found in certain tissues, such as certain parts of the liver, some exocrine glands, and very classically the kidney glomeruli. And again, I believe Dr. Tamagnini talks about the kidney in his lectures. Finally, we also in some tissues get what we call discontinuous capillaries. These have much larger gaps, and this allows passage of water, of all water-soluble molecules, including larger proteins which couldn't pass through a fenestrine. These are not found in many tissues, but they are found in some parts of the liver where you have sinusoids, also some parts of the bone marrow and the spleen. And we do mention an example of sinusoids in the PM1 liver lecture. And this is just a discontinuity in the wall of the capillary, which makes them very, very leaky. Now, across all of this, you've got to remember that it is pressure gradients that determine fluid exchange. Okay, so it's pressure gradients of the fluids that determine how the fluids are exchanged amongst our body. And this involves a variety of forces called Starling forces. And in the PowerPoint presentation, at the end of this presentation, I provide a link where you can click on to learn more about these Starling forces that I just simply do not have time in this lecture. It's worth bearing in mind that in terms of exchange, lipophilic chemicals can diffuse freely along concentration gradients, so are not really part of this. However, solutes require transport, or they move with the fluid movement. So most solutes will either be done by active transport or move with the fluid. The direction of fluid movement depends on pressure gradients, and those pressure gradients include the pressure inside the capillary, the pressure of the interstitial fluid, the fluid that surrounds the cells, and the pressure of the capillary is much higher than the pressure of the interstitial fluid. And the other thing that is always important to remember, contributing to these pressure gradients, are the oncotic pressure of the plasma. And the oncotic pressure of the plasma is the osmotic pressure produced by the large protein molecules found in the blood plasma. Now, there are proteins found in the interstitium, but again, that is lower than the osmotic pressure of proteins in the plasma. So the interstitial osmotic pressure is lower, and they're often represented by these various symbols. I'm not going to go into them into too much detail, but it's worth bearing that in mind. It'll be acts as a guide for the next slide. So in general, hydrostatic pressure is greater than the interstitial pressure. So here in this diagram, we have blood flowing from the arterial end to the venous end. Okay, so what we get is net filtration. So we get net movement of fluid all across the capillary, both from the arterial end to the venous end. So even though the pressure at the venous end is lower, we still get movement into the interstitium. Okay, hopefully that makes sense. So this diagram will show you the pressure, but the net pressure is always greater across from the capillary compared to the interstitium. So you will always get fluid leaving the capillary beds. Okay, so that means the body has a problem. It means unless this fluid in the interstitium is somehow then able to be removed, we will get fluid retention. And a lot of diseases do involve fluid retention. It's how you get swelling that's associated with many diseases. But the body has a way of getting rid of this excess fluid. And that getting rid of this excess fluid caused by the filtration pressure is one of the major roles of the lymphatic system. Now, I'm not going to go into the lymphatic system in any great detail. It's another circulatory system of your body, and you can imagine of it as the interface between the circulation and the immune system. Now, lymphatic vessels drain fluid from tissues back into the blood. So here we have a blood capillary, the interstitial fluid, and we have a lymphatic capillary. So the lymphatic system has discontinuous capillaries at this region, but it also has a valve system which encourages slow flow of fluid. Now, all fluid removed from the interstitial fluid passes via these lymphatic capillaries into the lymphatic veins, and then must pass through a lymph node before the fluid is returned to the venous circulation. Therefore, we're removing the fluid from the interstitial fluid back into the blood flow. Here in the lymph node is, and often this is a site of inflammation, that's where it'll be monitored by the immune system. White blood cells often penetrate into the blood supply. So to summarize, I've already mentioned why we have a circulation system and what it's for, and how that we have highly specialized arteries, veins, and capillaries. We've mentioned that the capillaries are the major site of solute exchange, and how excess interstitial fluid is removed by the lymphatic system. I'm not going to go over the learning outcomes here, but it's worth bearing in mind that all this material is probably best read in the further reading, along with the next couple of lectures, in chapters 20 and 24 of Pocock and Richards. And I would say that the material in these chapters is examinable as well as this lecture content. This resource also allows you to self-test these key components, and you can look at these resources by going to the online resources for Pocock and Richards by following this link, which will be available in the PowerPoint presentation. I mentioned previously there's a video on Starling's forces which I didn't have time to go into, and that's ran by Nottingham University, and the link again will appear in the PowerPoint slides accompanying these.